So the next section deals with muscular contractions. So last section, kind of looking at the very small parts inside of the muscle. Now what happens as your muscle actually contracts? So muscle contraction has several parts to it, and what it basically does is result in those sarcomeres, those bands or blocks, are going to shorten, and when they shorten, that pulls the muscle. And that'll be important. So for example, when we talk about origin and insertion, if I'm looking at my bicep muscle and this moves, I know that the insertion of my muscle is going to be on the end that moves something. So when my bicep contracts, it pulls this arm up. So the origin is going to be at the non-moving part. The insertion is going to be at the moving part of the muscle. So, myosin and actin again. Myosin has two twisted strands, and here again we'll look at the diagram. They have globular or kind of rounded heads at each end. Actin is an actual protein, and it's put together into those twisted filaments with myosin. So what happens when your muscles contract is those two are going to come together. Tropomyosin and troponin are the two proteins that we said were associated with your actin or your thin filaments. So here's the picture. Myosin, here's those globular heads that they're talking about that come off to the outside. Actin, quite honestly, looks a little bit like DNA. It's twisted. It's kind of like that twisted ladder shape. So I have my actin filaments and I have my myosin filaments with the heads that are going to extend out from the main filament on the inside. Oh, the other thing, when I'm looking at actin, my tropomyosin are these long, skinny tubes that are on the outside, and then troponin is going to be found on the inside. So what happens is we have a model that we look at when we look at muscular contraction, and it's called the sliding filament model. So my myosin's going to slide and my actin's going to slide. So this myosin head builds a cross bridge and attaches to a bonding site for the actin. And when it bonds, it's going to pull on the actin filament, and then it's going to release and it's going to pull, and that's basically how your contractions are going to work. Energy is needed for all of this contraction. And the main source of energy comes from ATP. Big biology word. ATP is a source of energy. Mitochondria builds a lot of ATP. When ATP is converted to ADP, energy is released. And then that energy is used to make these cross bridges fire or actually work. ATPase is an enzyme. Anything that ends with ase is typically an enzyme. That's going to help to produce ATP and help your sliding bridges to work. Here again, like I said, the main neurotransmitter in making your muscles contract is acetylcholine. So that's the main neurotransmitter that's going to be released that's going to stimulate a muscle contraction. Protein receptors in your motor end plate part of the muscle are going to bind to or accept those neurotransmitters. And then there's going to be a huge release of sodium ions. Sodium is very, very important for muscular contraction. Electrolytes, Gatorade, all of those are based on this fact. In order for your muscles to work well, you need electrolytes. particularly sodium. When sodium enters, you're actually going to have a charge because sodiums have a positive charge. There's your chemistry lesson, Na+. So these charged particles are going to basically create an electrical impulse. And that impulse then spreads over the surface of your muscle, and that impulse is going to cause your muscle to contract. Once your muscle receives that impulse, the sarcoplasmic reticulum releases calcium. So basically I'm getting a sodium-calcium exchange. So sodium is coming in, and then calcium is going to be released. Here again, this calcium is an ion. It's a charged particle. High concentration of calcium enters the sarcoplasm. It's going to interact with your filaments, and the myosin is going to be exposed. And you'll see more of that. Myosin cross bridges are now going to bind. They're going to attach to your thin filaments. They're going to pull them together, and your sliding reaction is going to happen. We'll get to the picture and it'll make more sense. After the nervous system the impulse has been received, acetylcholinesterase is what's going to get rid of your acetylcholine. So it's like the anti-part of it. In order for your muscle to relax, I need to get rid of the acetylcholine. Contraction continues until my acetylcholine has been decomposed and the nerve impulse stops. Once it stops, calcium returns to its normal condition. It goes back into being stored. The links are broken, so that sliding filament's going to come back out, and they both return to their original position. So here's where you actually see what's going to happen. So if I start with number 1 up here at the top, I have a relaxed muscle. I have ADP and my extra phosphate. I have my thick filament. I have my thin filament. And then what happens for a muscle contraction, calcium is going to be released, and it's going to actually bond to part of your thin filament. Tropomyosin is going to move aside, and then what's going to happen is my thick filament bonds to my thin filament. So this globular head that it's talking about actually attaches to the thin filament. When that happens, it's going to attach, these are going to bend, and they're going to pull that thin filament closer. ATP binds to the myosin, breaks that connection. Once the connection is broken, things are going to go back towards normal, and you're going to go back to muscular reaction, or more calcium will come in and start it all over again. So this is what it looks like when I'm looking from a side view. If I'm actually looking microscopically, and I like this diagram better. What happens when you're relaxed is my thin filaments are here, my thick myosin filaments are here, they're not attached. When they contract, I have this binding site where they have attached. Your thick filament pulls, and it's going to pull the thin filaments in, and it's going to get much shorter. So those sarcomeres get shorter because they have pulled these in together. So now my thin filaments have moved in closer and pretty much surround my thick filaments. And I can see that same thing. So here's that Z line that shows where your sarcomere is at. As it starts to contract, they get a little closer, full contraction. My Z lines are way in here. So I can see that entire block has shortened a large amount. You can actually measure, and we did this in college. I don't do this in lab in high school, But you can measure the length of your muscle when it's relaxed, and then measure the length of it when it's contracted, and see how much it actually shortens when those filaments slide over each other. I'm not going to read through this. This is in your book, it's in your notes. It goes step by step over the actual steps that happen during a muscle contraction. So basically it's summing up the last few pages of notes. Sources. We're about done. A couple more slides. In order for my muscles to contract, I have to have energy. Main source, ATP, which is built by your body. ADP and P come together, I have ATP. It's limited in supply, but your body can regenerate it quite quickly. So the initial source is going to be ATP that's stored in your muscle, and that does not last for a very long period of time. Creatine phosphate is also stored in your body, and it's going to store excess energy that the mitochondria has made. So creatine phosphate is also a source of energy, and it can help make more ATP. So whenever the supply of ATP is sufficient, it's good, it's enough I need, creatine phosphokinase, another enzyme, promotes the synthesis of creatine phosphate. Because I'm using my ATP, my extra energy is going to be stored as creatine. As ATP is used up, so my muscles are working and using ATP, then that energy from creatine phosphate can be transferred to make more ATP, and that's going to produce more energy. As the supply of creatine phosphate declines, so I've used my ATP, now my creatine levels are decreasing. The cell must rely on cellular respiration, which uses sugar, glucose that is stored in your body. So creatine supplement. Does anybody know someone that uses creatine supplement?
Shane Wilson.
Okay, it's common, and creatine supplement is not a bad thing. However, too much is not good for you. Too much is going to basically be excreted through your body. So if you want to take a creatine supplement for typically to build muscles, things like that, if it tells you to take 1 tablespoon and you think 5 would be better and will build more muscle, that's not right. Well, the recommended dosage is typically pretty high anyway. So more is not better. Your body can only use so much, and the rest will it'll not be used at all. More is not better. So here's just a quick diagram. If I have enough ATP for what my body is doing, my creatine is going to basically be stored. This is not the best diagram ever. But ATP is high, my creatine is going to be stored. When ATP is low, my creatine is actually going to be used. So the arrow at the bottom shows that I'm going to use creatine. The arrow at the top shows that I'm using ATP. So ATP first, creatine second is basically what that diagram shows. And I'm going to stop there because the next section will deal with muscle fatigue, and I want to keep that separate.